Hey, what is going on? It is me, and it is Wednesday, March 30th, 2022. The year is flying by. We're almost at the 1 quarter mark, and I think we've done a lot in biochemistry right now. We're working our way through gluconeogenesis, which yesterday I kind of gave a brief introduction to, and yeah, it's quite the beast, actually. It's funny because there's less overall steps involved in gluconeogenesis versus glycolysis, as you can see on this diagram here. But because gluconeogenesis, recall, does for 7 of the 10 steps of glycolysis the same thing in reverse, right? Remember, there's 7 reversible reactions. There's reaction 2, reaction 4, 5, 6, 7, 8, 9. Those are reversible, and therefore gluconeogenesis goes backwards through the reactions, the reversible reactions. And remember, these are reversible because these are not the so-called committed steps, and when we get to the committed steps, those reactions are not naturally reversible. Remember, the goal is to form glucose, right? Gluconeogenesis, new glucose creation, but from pyruvate. But so today I'm going to try to go through the first of the non-committed steps, or first of the irreversible steps, all the way on the bottom where we have pyruvate, and I'm only going to go through half of that first step because that first step actually consists of 2 sub-steps. Okay, so I'm going to try to go over the first sub-step of that reaction, and the reason why is because we need to understand, like, the good thing about going through gluconeogenesis and the fact that we went through the Krebs cycle earlier was that we get to see the use of oxaloacetate, right? Remember, oxaloacetate exists within the cell, in the cytoplasm, and the mitochondria. But oxaloacetate, if you remember the malate shuttle that we talked about in the past, oxaloacetate stays in the mitochondria, and it has to go out of the mitochondria because in order to complete gluconeogenesis, that's where gluconeogenesis continues in the cytoplasm. So to go out of the mitochondria, oxaloacetate, which is a main intermediate of this whole process, needs to be converted to malate through the utilization of malate dehydrogenase, and then that is first reduced, and then malate is transported out of the mitochondria. There's just certain transporters, remember, which are essentially molecular exchange transporters we talked about in the past. The molecular exchange transporters exchange a malate, and the oxaloacetate then leaves, the malate leaves into the cytoplasm, and then that converts back to oxaloacetate, so the rest of the reaction can occur. It seems kind of excessive, honestly, but evolution probably has a good reason for doing it this way, most likely energetic in nature, right? Because at this level, molecular level, we're always trying to reduce energy, which is the whole reason why we're doing gluconeogenesis in the irreversible steps through a fairly complicated reverse reaction. Okay, so why did I talk about oxaloacetate, right? And why did I talk about this guy here? And just remember, oxaloacetate has these oxygens everywhere. Because oxaloacetate is the intermediate that's formed essentially from pyruvate carboxylase. Okay, so let's recall what we're trying to do with gluconeogenesis. We're trying to get from pyruvate to phosphoenol pyruvate. In order to go from pyruvate to phosphoenol pyruvate, right? Remember, in order to go from phosphoenol pyruvate to pyruvate, we need pyruvate kinase. But in order to go in reverse, we first need to convert to oxaloacetate, and then we need to convert oxaloacetate and we do that through the use of pyruvate carboxylase, which exists in the mitochondria. We need to convert oxaloacetate to phosphoenol pyruvate through the use of PEP carboxy kinase. carboxykinase I'm going to try to avoid today. But talking about pyruvate carboxylase. So pyruvate carboxylase exists within the mitochondria. It's a very interesting and complex enzyme. And essentially, one thing you have to know about it is that pyruvate carboxylase, it can only be activated to convert pyruvate to oxaloacetate. It requires several things. It requires acetyl-CoA, first of all. So our good friend acetyl-CoA, which is created from the transfer phase in the mitochondria, those are acetyl-CoAs. Instead of being involved in the Krebs cycle, can be involved in gluconeogenesis. Okay, so things kind of tie in together through all of these. Acetyl-CoA is bound to pyruvate carboxylase, and then pyruvate carboxylase, okay, uses a molecule called biotin, right? And biotin is an interesting molecule. You obviously have seen it. You probably see it in the store, but pyruvate carboxylase, without going super into it, has a biotin carboxylation domain. Okay, so this biotin carboxylation domain requires that you bind CO2 to it. Okay, so this is interesting because biotin itself, again, it's a naturally occurring molecule that occurs and is linked to this pyruvate carboxylase, right? It will bind to CO2, but that's not the whole story. It needs to bind to CO2 in order to form oxaloacetate, and then you have biotin. That the biotin is unbound. So oxaloacetate is basically the combination of CO2 and okay, so let me step back a little bit. So CO2 by itself, right, doesn't exist in your blood. Okay, CO2 does not exist in your blood by itself. Okay, it can't, because it's polar. It does in small amounts, but it's a non-polar molecule, and so in order to exist in solution with H2O, it actually has to be converted to bicarbonate through the use of an enzyme called carbonic anhydrase. And how carbonic anhydrase works and where it works, that's a long story in itself. But let's say CO2 is dissolved in your blood via this use of this enzyme, and then carbonic anhydrase does that, and then you can combine, you can allow the biotin to bind to the bicarbonate and form carboxybiotin. Okay, so biotin and carboxybiotin can then go through the process of, there's a lot of biochemistry that's involved here, and I'm going to go into carboxybiotin biochemistry at some point in the future, but at this point we'll just say that then the enzyme's action, remember the enzyme that we're talking about is pyruvate carboxylase. Pyruvate carboxylase, and to note, these reactions are still endergonic reactions. They still require a fair amount of energy to occur, so you're asking, why is it worthwhile for these endergonic reactions to go on, especially if they're more complicated? Well, these endergonic reactions can be catalyzed by co-reactions. These co-reactions, the first one, in the presence of ATP, in ATP phosphate extraction to turn ATP into ADP plus P, that reaction will occur at the same time pyruvate carboxylase does its work to convert the pyruvate plus carbonate to oxaloacetate. Okay, so the reaction becomes, in effect, exergonic. Okay, so you release energy in this reaction. So this will, cause frictional heating in the system, and the energy of overall system will decrease via this reaction's usage. Right? The reaction will be putting energy into the system. Okay, so the Gibbs free energy, another cleaner way of saying it is the Gibbs free energy for this reaction in the presence of the ATP dephosphorylation will cause this reaction to move forward. So I basically just wanted to talk about that, because the next step after the formation of oxaloacetate and the transfer of oxaloacetate into the cytoplasm and the conversion of the malate into oxaloacetate in the cytoplasm. Remember, that's how we transport oxaloacetate out of the two layers of the mitochondria. At that point, then we need to involve the second reaction, or the second enzyme, which is PEP carboxykinase. And PEP carboxykinase is also a reaction that under Pepsi carboxykinase's action is also a reaction which is androgonic under normal body conditions, and it's exergonic with the help of another co-reaction at the same time that occurs. So we'll go into that next time, and I'll also go into the reverse step 3 of glycolysis, which is very important to understand. The fructose 1,6 bisphosphate reaction requires fructose 1,6 bisphosphatase, and it's a whole different complex step in itself. Okay, so thanks for watching. I will see you later.